During the electron transport system, here the protons are also translocated from mitochondrial matrix to intermembrane space. So this translocation of protons, that is the source for proton concentration is important for the ATP synthesis. So the proton concentration gradient, the proton gradient is established in the intermembrane space. In this space, the protons are here accumulated or deposited. So here from this intermembrane space, again the protons, all these protons are moving from mitochondrial matrix to intermembrane space. So after movement here, high number of protons are present in the intermembrane space. So all these protons are again they try to move mitochondrial matrix. But here the membrane is impermeable. This total membrane is impermeable to protons. So how the protons enter? So generally the protons, based on the proton concentration gradient, based on the concentration gradient, the protons are moving from high concentration to low concentration. Here high concentration of protons is there, but here low concentration of protons. Here the membrane is impermeable. So these protons are moving from this space to here mitochondrial matrix through this complex. This complex is called as F0 F1 complex or ATP synthase F0 F1 complex or ATP synthase or elementary particle also. So it is F0 F1 complex. So in the F0 F1 complex, here one stalk-like structure is present and one head-like structure. So the stalk-like structure is called as, it is F0 part, and it is F1 part. Head-like structure is head-like part it is, and stalk-like structure is F1 part. So F0 part is embedded within the membrane, and F1 part or this head-like structure, it is exposed to mitochondrial matrix. It is present in the mitochondrial matrix. So only this complex is here permeable to, or it is responsible for the proton translocation from here all these protons. So the protons are moving from intermembrane space to mitochondrial matrix. Every two protons, if two protons are translocating or entering through the F0 F1 complex, here one ATP is produced. For two protons, one ATP. Here F1 part or a head-like structure, it is having one, it is the site for ATP production. So this F1 complex is, here it is, when the protons are moving from intermembrane space to mitochondrial matrix, the F1 part is rotating. So some energy is released. That energy combines the ADP and the phosphate. So ADP, adenosine diphosphate, and it is combined with inorganic phosphate. So ATP, adenosine triphosphate, is synthesized. So here two protons translocation, one ATP. If two protons are moving, one ATP synthesized. If four protons are moving, here two ATPs. For six protons, total 6 protons are moving, here 3 ATPs are synthesized. So here when mitochondrial NADH2 is oxidized, the electron movement is like this. Up to oxygen. Here when these are moving, 6 protons are translocated. 2 protons here, here, here. So total 6 protons. When 6 protons are moving, here 3 ATPs are synthesized. So it is equal to 3 ATPs. When FADH2 is oxidized, here the electrons are moving like this, and here 2 protons are translocated, here also 2 protons are translocated. So 4 protons are equal to 2 ATPs here. So that's why the FADH2 is equal to 2 ATPs. Next, when glycolysis NADH2, when glycolysis NADH2 is oxidized, here 2 protons are moving from here to here, and here 2 protons are also translocated. So total 4 protons are translocated during the oxidation of glycolysis NADH2. So 4 protons are producing here 2 ATPs here. So glycolysis NADH2 is also equal to 2 ATPs. So here glycolysis NADH2 is giving here 2 ATPs, or one glycolysis NADH2 is equal to 2 ATPs, and mitochondrial NADH2 is equal to here 3 ATPs. FADH2 is equal to here 2 ATPs. So during the here electron transport system, some protons are also translocated from mitochondrial matrix to intermembrane space, and these here protons are accumulated in the intermembrane space. So the proton concentration gradient is established in the intermembrane space. So the high concentration of proton is present in the here intermembrane space. So they try to move inside. So while they are moving inside, here ATP is synthesized. For every 2 protons translocation from intermembrane space to mitochondrial matrix, here 1 ATP is synthesized. 4 protons are translocating, 2 ATPs. If 6 protons are entering into the mitochondrial matrix, here 3 ATPs are synthesized. It is a oxidative phosphorylation. So here in the presence of oxygen and during the oxidative reduction stages or oxidative reduction phases, here the ATP is produced. ATP is synthesized in the presence of oxygen as well as in the oxidative reduction reactions. So it is here it is called as oxidative phosphorylation. So this oxidative phosphorylation was explained by Peter Mitchell. Peter Mitchell explained the oxidative phosphorylation by chemiosmotic theory. So the ATP production, it is based on the Peter Mitchell's chemiosmotic theory. So one glucose molecule, it is giving a number of ATPs. So how many ATPs are synthesized, how many ATPs are produced during the aerobic respiration. So we will discuss in the next topic. It is about aerobic respiration, 3 steps, and oxidative phosphorylation. It is the 4th step. So ATP production during the aerobic respiration, here ATPs are synthesized. So it is about oxidative phosphorylation.